Well, you're in for a real treat because we're going to talk with Brad Marshall again, who is essentially a molecular biologist who's passionate about understanding and reviewing the medical literature and engages in many areas of research to help our understanding of food physiology and biology so that we can change or make better choices to help improve our body's ability to create cellular energy. And one of them are pretty surprising things. And, we're typically taught that monounsaturated fats are relatively healthy and maybe even beneficial. And there may be some benefit from them, but the vast majority of people, there's a series of complex enzyme reactions that occur that convert the saturated fat in your body to these monounsaturated fats. And when that ratio gets disturbed, it can really radically decrease oxidative stress and inflammation in your body and produce something like a hibernator experience or state that is also called torpor in animals for sure, but we can reproduce that in humans. So it all has to do with some of the changes that occur when you're eating certain types of fats. And we're going to go into this in detail so that you can take advantage of this information and make choices that will likely improve your body's ability to create the energy you need to provide all the changes that your body requires to move your body in the right direction towards health. Because if you don't have cellular energy, your brain's not going to work, your heart, your liver, your kidneys, and your gut. And your gut, if your gut doesn't work, you're in for trouble, because that really is one of the cores of why we're staying healthy, because there's trillions of bacteria in your gut. And if you're not providing an environment where the right types of bacteria can grow and nourish and produce the biological molecules your body requires to optimize your health, then you're going to be in trouble. And yes, you can throw ostensibly healthy symptomatic supplements and Band-Aids in there, but more important is to understand the reasons why those changes occurred and address them at a fundamental level. And one of the major ones, of course, is the foods that you're choosing to eat and its consequences on your body's ability to produce the cellular energy or increase your metabolic rate. And we're going to dive into that and provide an exciting resource for you to consider to learn more about how you can optimize your body's ability to produce cellular energy.
In the last, of course, in the last 100 years, we've eaten more and more and more of these unsaturated fats. You've talked a lot about concerns with polyunsaturated fats. And what I see when I look at it is the linoleic acid is sort of okay if it stays as linoleic acid. But once it starts getting converted to oxidized fats, that's when it becomes a real problem. If you trigger the aryl hydrocarbon receptor, you get dysregulated gut microbiome. And so if you're oxidizing a lot of polyunsaturated fat, you activate the aryl hydrocarbon receptor, and all of a sudden you have overgrowth of the bad gut bugs, and you'll have a decrease in the good gut bugs.
Welcome, everyone. Dr. Mercola helping you take control of your health. And today we're in for a real treat with the repeat return visit of Brad Marshall, who is an extraordinary individual, one of the most accomplished reviewers and readers of the scientific literature, specifically molecular biological literature, which is one of my favorite areas of diving into because it helps us understand biology, how it works. And if we understand how it works, we can use that knowledge to help us make better choices. And that's what Brad's going to do. He's going to help us dive deep. And remember, you can always hit rewind on this and listen to it a few times. And if you're really hard pressed to understand what some of the concepts are, let me encourage you to consider copy and pasting the transcript and putting it in ChatGPT and tell it to dumb it down for you and make it simpler. And it will do that. And if it doesn't do it the first try, keep on doing it because it's an infinitely patient teacher. And why is that so important? Because this is really the keys to your health. If you understand these foundational concepts, you can make choices which will move you out of disease and towards health because your body wants to be healthy. It was designed to be healthy. That's what it's supposed to do. It's some delusional belief system to think that we were designed to be towards disease, and the only solution to that is to pop a pill to somehow magically turn that around. That is the furthest from reality that you can ever imagine. So with all that said, I also want to acknowledge that I've admired Brad's work so much that I've hired him to be a research director for me. And he is actually, we'll talk about this. We're putting together some tests to measure the redox potential of your body. And these tests are not commercially available. They really do not exist anywhere outside of some exotic tests in LabCorp and Quest, which are very difficult to get and almost always collected improperly. So the results are questionable at best.
Yeah.
We'll talk about that in a bit. So with all that frame, we're going to talk, well, one of the things I want to focus on this, maybe we can open up with this, is because we left on the last conversation about reductive stress. And if you haven't seen that one, you're going to want to view that one again, because it's worth repeating, because it's such, this surplus of electrons that we get that accumulate in our mitochondria as a result of impaired mitochondrial function is really the core of almost all disease that we have. So it's why it's important to understand reductive stress, because if you understand it and you know how to remediate that, you can literally create magic in your body. So it's a lot of exciting preface to this. So with all that said, welcome and thank you for joining us today, Brad.
Yeah, thank you. Yeah, and I think also I'm very excited to be to get some research done, because I think we can. We're gonna move the ball forward. This
big
is
time. Big
We're
time.
gonna go. We're gonna move fast. I think we're way ahead of the curve, and it's not going to take us long to start to show that. So I'm really excited.
We don't want to disclose what our research areas are, because we're going to give people we want the unfair advantage.
Right. And thank you for the opportunity. Very excited about that. And but yeah, I 100% agree that reductive stress is essential. Is at the root of all of the problems that we're having. Like you say, this buildup of extra electrons, which pile up in places like NADH. Right. And we went through that in the last episode. So check out that episode. We talk about that. But I think in this episode, right, we want to get into a little bit. I'd like to think a little bit more about the kind of like why do we get into reductive stress? And I mean,
yes,
not only from like, you know, what did we do wrong? Because I actually don't. One of the things that I think why I come up with different ideas than some people is I don't really think about metabolic syndrome as a disease so much as an evolutionary process, right?
Mm-hmm.
And as I've mentioned, you've probably seen on my videos, that process is called torpor. And you know, animals, as they've navigated the globe over the past 500 million years, end up in all kinds of niches. And so there are times when you want to have a hot-blooded metabolism, and there's times when you want to be able to shut that metabolism down. And so in my mind, when we talk about metabolic syndrome, we've got a mismatch of we're turning on these systems that are sort of designed to operate in times of in times of shortage, which might be winter. Sometimes it'll be dry seasons in the tropics. Lizards do a thing called estivating through the summer, right, where they shut down their metabolism. People think about hibernation, of course, as being a cold weather thing, but animals also will hibernate or estivate through the dry season a lot of times. And so this is very much more broadly, this alternate metabolic state, which is defined by, as we talked about, if you look at animals as they approach, there was a great study in grizzly bears, actually, and they looked at the difference between grizzly bears in June, when they're in the active part of the season, and October. And you have to go into the supplementals of the paper to find the gene expression. But what they showed is that in October, when the bears are fattening, and they're gaining weight and putting on all this fat, they have pretty massively increased expression of all three of the desaturase enzymes that we talked about last time: delta 6 desaturase, delta 5 desaturase, and delta 9 desaturase. And the amount of monounsaturated fat in the bear is rapidly increasing as as they approach winter, because um, that's kind of the end result of all of these pathways, is the accumulation of monounsaturated fat. And the reason that the monounsaturated fat is in charge of this shift in metabolic state towards torpor is that animals can't make polyunsaturated fat. So if you have to rely on, and so if you have saturated fat, you're going to have a high metabolic rate. You are not typically going to be in reductive stress. Natambi showed this in the lab with mice. He showed that in mice, if they don't have this desaturase, this SCD1 that's converting saturated fat into monounsaturated fat, they have very low NADH to NAD plus ratio. They're not in reductive stress. They don't have this buildup of electrons. They stay lean, and their metabolic rate is 40 or 50% higher than a mouse that has this. And they can reduce the or they can increase the NADH levels and put those mice into reductive stress if they feed them a lot of unsaturated fat, right? So they showed very closely how tightly this idea that saturated fat is connected to a high metabolic rate and not being in reductive stress. And if you have a lot of these unsaturated fats, you're going to have low NAD plus availability, and you're going to have lower metabolic rate. And so that's a really nice experiment that they did. And you see what the bears are doing as they go into winter is they've increased all of these desaturase enzymes. And so they're desaturating their body fat to help them store fat and to get ready for the oncoming winter, right? And since animals can't really make polyunsaturated fat, the end result of that process is actually monounsaturated fat. And that's what you see in bears, but you also see this in echidnas, which are spiny anteaters, and they live in Australia. And that's a very ancient mammal, right? And we think the first mammals ate bugs, insects. And so when you see an anteater, I did a video where I said, don't eat the cold weather ants. There's a study in Brazil. They looked at the fat composition of Brazilian ants, and Brazilian ants are very, very saturated. Then there's another study. They looked at German ants. German ants are almost all monounsaturated fat, right? So if you think about mammals, they're moving around the world, and a lot of mammals are very widely dispersed. So you have black bears that live in Canada, and you have black bears that live in Florida, right? And the black bears in Florida don't hibernate nearly as deeply or as long as the black bears that live in Canada. And so one of the signals, I argue, that is telling the animals how torpid they should become, how much they should lower their metabolism, how much they should turn on all of these desaturase enzymes, is the fat that they're consuming. And since the earliest mammals were insect eaters, I think that the fat composition of insects was probably this very early signal to the mammals. Should we have a hot-blooded metabolism? Do we live in the tropics where the ants are full of saturated fat, as was shown in Brazil, or do we live in a northern country like Germany where the ants are made of monounsaturated fat, right? Because the ants can't make polyunsaturated fat either. And so they're accumulating this monounsaturated fat. And if the anteater or other early mammal comes and eats those ants, that's a sign that, winter is coming.
Yeah.
Fatten up, right? And so I see that in the last 100 years, we've eaten more and more and more of these unsaturated fats. And of course, the polyunsaturated fats contribute to this process as well, because the delta 6 desaturase is the limiting step in the oxidation of the polyunsaturated fats, right? And so I know on your show, you've talked a lot about concerns with polyunsaturated fats. And what I see when I look at it is the linoleic acid um, is sort of, it's still not my favorite fat, but it's sort of okay if it stays as linoleic acid. But once it starts getting converted to oxidized fats, that's when it becomes a real problem. And that delta 6 desaturase enzyme is the thing that gets upregulated in the fall in the grizzly bears and in other hibernating animals. It's been shown. And that is the thing that takes those polyunsaturated fats coming into the diet, and we start oxidizing them, and um, we start making 5 heat and 12 heat and 15 heat.
Well, it's not the delta 6 that does that. Delta 6 makes additional unsaturated bonds.
Right.
So it goes to, instead of 2 double bonds, it might be 3 or 4,
Right.
like in DHA.
Correct.
Yeah.
Correct. But what happens is that first, when you add that first double bond to linoleic acid, that's kind of like the gating factor. That's the limiting step, because once you add that third double bond fairly quickly, in general, the fourth double bond gets added, and then that linoleic acid becomes arachidonic acid. And arachidonic acid usually winds up in your cell membranes. And then and there's a lot of these enzymes, we make a lot of other enzymes that oxidize the arachidonic acid, and arachidonic acid is their favorite target. And so in all your cell membranes, you have a lot of this arachidonic acid, and there's an enzyme that comes and releases the arachidonic acid from your cell membranes. And then there are other enzymes that oxidize that arachidonic acid into the 5 heat and the 12 heat and the other oxidized polyunsaturated fats.
Let's stop here for a moment, because when I wrote my paper on linoleic acid review, and there were some of the peer reviewers who sent me studies to evaluate, and they had a pretty strong argument that there's a belief within many people who dislike linoleic acid as a food source, to justify one of those reasons why not to like it is that it's converted, like you said, from delta 6 to arachidonic acid. And arachidonic acid is a precursor for many of the inflammatory prostaglandins, eicosanoids.
Right.
But there were some pretty elegant studies, and I don't remember the details because it's been about 2 years since I did the review, that were presented that convinced me that really isn't the primary mechanism at all. That's not. It's really limited. The inflammatory prostaglandin release from linoleic acid is a minimal reason why it causes damage. That is not the main reason. Is the one you quoted earlier. It's this conversion, not by delta 6, but by other oxidants or free radicals into these oxalams, for short, oxidative linoleic acid metabolites, which are very pernicious, and that's what causes virtually all of the damage.
Right. I agree. And I'll tell you the reason, right? And so that kind of nicely segues us into, so what do these oxalams do? And they do a lot of things, but one of the things is we have these things called nucleoreceptors. And again, if we think about this evolutionarily, nucleoreceptors are the things that differentiate animals from all the other things on the planet. So if you look at our closest related single-celled organisms to animals, they don't have nucleoreceptors. And then when you look at
they don't have a nucleus, because they don't have a nucleus.
Well, no, but I'm talking about eukaryotes, single-celled eukaryotes that are similar in a lot of ways to very small multicellular animals, but they don't have nucleoreceptors.
Okay.
Then when you look at a sponge, which is the most ancient, we think, is the most ancient sort of multicellular animal, the sponge has a vitamin A receptor. And that's the first sort of nucleoreceptor that we see, and it is involved with cell differentiation and kind of how multicellular things communicate with each other. And we've got a bunch of these other nucleoreceptors, and you guys will know a bunch of them: the vitamin D receptor, thyroid hormone receptor, the estrogen receptor, the androgen receptor. And so when you think about your endocrine system works entirely or almost entirely through signaling these nucleoreceptors. And the nucleoreceptors are in all your tissues. And what they're doing is they're recognizing certain signals, right? Which, like I say, could be thyroid hormone, could be vitamin D, or they can be things that are coming in from the extracellular environment. A good example of this is BPA, the thing that's in plastic, triggers something called SXR, also called PXR. That's a very famous example of a thing that we're exposed to all of the time, right? And they say it causes endocrine disruption, and that's because it is triggering certain nuclear receptors.
Well, it should
And
also
so,
trigger the estrogen receptor too. It's a
right,
xeno
yeah, they
estrogen.
trigger a bunch of different nuclear
Yeah.
receptors.
Yeah.
I think that's right. And so, but these nuclear receptors are essentially looking. They're always looking for signals, and the 5-HEAT and the 12-HEAT specifically are they came from the linoleic acid.
Mhm.
They will trigger they'll trigger the something called the aryl hydrocarbon receptor, and they'll trigger PPAR alpha. And if you look at those two things, those I believe are hugely those are sort of in control of shifting that metabolism towards a more torpid metabolism. And it seems like they're controlled by some of these oxidized linoleic acid metabolites. And so once you start to trigger those things, right? And so you have this path where um, as certain environmental signals come in, and those can be things like eating too much unsaturated fat, but it also can be things like BPA or environmental toxins, which we're of course exposed to all of the time.
Mhm.
You start to trigger these kind of higher level control nuclear receptors, and the aryl hydrocarbon receptor is particularly interesting. And I'll tell you one, the aryl hydrocarbon receptor, if you give a mouse a drug that strongly activates the aryl hydrocarbon receptor, it shuts down the circadian rhythm of the mouse. So the aryl hydrocarbon receptor controls your circadian rhythm. And so, people who have metabolic syndrome have issues with sleep, and obviously, this is a big category, right? But so that's one thing, is the sleep. But the other thing, that if you trigger the aryl hydrocarbon receptor, you get dysregulated gut microbiome. And so if you're oxidizing a lot of polyunsaturated fat, you activate the aryl hydrocarbon receptor, and all of a sudden you have overgrowth of the bad gut bugs, and you'll have a decrease in the good gut bugs. And
Wait,
so,
no, stop there. That's I don't understand why that would be. How does the activation of the aryl hydrocarbon receptor catalyze the transition between beneficial and pathogenic bacteria?
well, I don't know if it's entirely known, um, but the AHR is highly involved in controlling the immune system.
Mm-hmm.
And so one of the things you see if you trigger the AHR, and I'm looking at my cheat sheet here because I always forget this term, but you see this increase in what are called myeloid-derived suppressor cells. And so these are cells, they're immune cells that come on, and they essentially, they suppress your immune response. They're immunosuppressive, I guess is the word. And so anytime you trigger the AHR, it's a huge controller of immune response. And so it can, I think a difference between myself, we talked about the repeat community, and one of the reasons this discussion came up, and I'm telling the audience here, is that so there's this discussion about, we've been talking about eating more carbohydrates and burning more glucose,
Mm-hmm.
and I'm a big believer in starch, and a
Yes.
lot of people in the repeat community don't like starch because they say that the starch will, and especially the things like the resistant starch, if it goes into your gut, it will trigger this growth of bacteria, and the bacteria release these things called LPS, lipopolysaccharide. The lipopolysaccharide can get into your bloodstream and cause all kinds of bad problems, and you definitely don't want a high LPS response. LPS does cause all kinds of problems, and so we have this debate about
Right. So this is where I wanted to weigh in. This is the why I had you back, because I thank you for giving me this opportunity, because this
Yeah.
is huge, and I want to take off and definitely engage in the conversation. But this is huge, huge, huge. This is the major point. Great admiration and respect for everything that Pete has done, but he didn't get this. He missed it, and almost everyone in the Pete community misses this, and it's a very important point. And your work really helped me understand the reason, because I've been studying his work for a year and a half, good year and a half, 18 months. And that's a lot of time for me. And I've now started to understand the flaws in the work. And it's related to what you're saying, this misunderstanding of the carbohydrates. They classify all carbohydrates or similarly. Starches are the same. What's not appreciated, I don't think it's as complex. You get into this, you talked about the aryl hydrocarbon activation and causing its secondary immune activation, which is responsible for the transition. That may play a role, but I doubt it. If it does, it's minimal. And I'll tell you, I'm going to share with you something that is not widely known, hardly by anyone. This is a real innovation. And there are some, actually, Ashley Armstrong, who's a mutual friend of ours, who just bought your hog farm yesterday, I believe. Mutual friend of ours who's just totally aligned, and I'm talking with her soon also. And she's been on the podcast before, but she knew this, and she found studies that help her understand this, but there's like a handful of people who understand this. So let me share. What is this? What is this? This is the fact, all relates to what your primary work is to mitochondrial function, the one we are in absolute complete alignment with. And then when your mitochondrial function starts to decrease, that's when the problem starts, and especially starts in the gut. One of the primary functions of the gut is to maintain an anaerobic environment. That is an environment without oxygen. And I'm not sure of the exact mechanism, but you need energy to keep oxygen out of there. And if that energy is not available, oxygen is going to seep in, and they're the primary bugs that are designed, the beneficial bacteria. Almost all these beneficial bacteria are gram-negatives, and they're called obligate anaerobes, and they do not have LPS or endotoxin in their cell wall. It's not there. But when they start to die, they leave us a little niche, and there's a principle in microbiology called competitive inhibition. So when the beneficial bacteria start to die, there's this niche that's available, and these other bugs come in, and these are called facultative anaerobes. In other words, they can tolerate oxygen and survive. And these facultative anaerobes, these are the bad guys. These are the pathogenic bacteria, and embedded in the cell wall of the pathogenic bacteria is LPS and endotoxin. Now, interesting, there's another little subtle point here. The primary obligate anaerobe is a species that was just discovered 20 years ago, Akkermansia. But it has a last name. It's mucinophilia. That sounds a little bit familiar. Mucin. What does Akkermansia do? It actually eats and makes mucin. What is mucin? It's the protective layer in your gut that smooths everything. It's like the barrier, the protective barrier. And when your Akkermansia disappears because you don't have enough cellular energy, because you can't maintain that oxygen gradient and exclude it from the large intestine, then you don't make mucin and you get leaky gut. That's when these, listen, that starch that you said that the Pete community is concerned about, they should be concerned. Why? Because just like metabolic, there's 95% of the population is metabolically inflexible. That's really clear from the enhanced data. And it actually was 93.1%. That was 2019 data, maybe 18. It's literally 6 years old. So that number is probably closer to 97% by now. So 97% of the people are not making enough mitochondrial energy. That means 97% of the people have an impaired gut microbiome. And so the Pete's community is valid for 97% of the people, because they have a disturbed microbiome, and that starch is indiscriminate. It's fertilized for any bacteria that's growing in the colon, beneficial or pathogenic. Since most people have a preponderance of pathogenic, it causes problems, and they stay away from starch. They should. They need to. Unless you have a healthy microbiome, I don't think starch is going to be beneficial for you. In fact, I would strongly advocate to avoid it. And listen to your body, of course, but my primary goal is to get your cellular energy up, and improve that microbiome first, and then you could take the starch, and then you go off to the races, because that is your data and research convinced me overwhelmingly, and I've radically changed my diet now. I'm taking 1.5 pounds of rice a day. I'm up to 600 grams of carbs, 600 grams, and much less fruit now than than rice carbohydrate, which is starch. And starch is one of the, it may not be obvious, but one of the primary components of starch is that it doesn't have fructose. It's fructose free. So
Yep.
I think, I was becoming disenamored more and more with Lustig and Richard Johnson, but I think that there's some elements of their concern that is valid. And you don't really, excessive fructose is not your friend, certainly if it's in process. And I think fruit is good, but you got to be careful. You don't want to go crazy with fruit. So I think starch is your ideal fuel. So that is the crux of the what I wanted to share, and I'll certainly let you chime in, but I think that is it. I don't think it's this aryl hydrocarbon activation, although it may play a role. I think it's minor. I think it really is the lack of cellular energy because of reductive stress, which we talked about last time, which is the single most important reason why we have mitochondrial dysfunction. And the primary contribution of this, we both agree, it's not only linoleic acid. I am convinced now, we're going to talk about this next, is that the monos are almost every bit as important. And you're the expert in that, or you're going to teach me. But I am now, we'll talk about it in a bit, but I am not a fan of monounsaturated fats anymore. And I assiduously avoid them, assiduously.
Yeah. And so to sort of piggyback off what you said there, I actually don't think that, so to me, this is all part of one sort of big, complicated, positive feedback loop, right? Mm-hmm. And so what you said, I think, you're absolutely 100% right. I 100% agree with you about the gut barrier function. Everything you said there, I think, is correct. But I also think that, there's a whole bunch of sort of aligned systems that are pushing us towards this outcome. And just to get back to real quick to the hibernating animals. When you look at the hibernating animals, when they go towards winter and you see all those desaturase enzymes are increased, their gut permeability increases. And so they take these squirrels in October and they check the gut and they put ions in them and see how many get through, as opposed to the same squirrel in summer. And so one of the tricks that the animal does as the hibernating season is approaching is they increase gut permeability, and they let more LPS in. That may also let more oxygen through. I'm not sure. And we talked in the last one, we talked about glycine, right? And so one of the patterns that you see in obesity in metabolic syndrome is you see that branched chain amino acids in your bloodstream increase and glycine goes down. And you can show in mice that if you supplement with glycine, it increases gut barrier function. And so you have this pattern going into winter where the AHR is activated and these desaturases are activated, and gut barrier function is decreasing and LPS is coming in. And again, that's contributing to the thing about the LPS is it causes release of something called CD38, and CD38 breaks down NAD+. And so anytime LPS is coming through, it's decreasing NAD+ availability, ultimately probably because of this enzyme CD38. But
it's one of the two biggest consumers of NAD in the body. There is PARP.
Right, yes. And the thing about that is, like I was saying before, all three desaturases run off NADH and not NAD+. And so the gut inflammation leads to increased activity of these desaturases, which leads to increased monounsaturated fat production, which continues to push you further in that direction of reductive stress, which lowers glycine, which makes the gut more leaky. It's all, I think it's all wrapped. To me, it looks like it's all wrapped into the same process, because of course, remember that if you're a spiny anteater living in southern Australia, your biological imperative is to fatten up for winter, right? And so these processes, they all work together, and they all work in these feedback loops, because if you're an animal in the right situation, you want to have a low metabolic rate, you want to have slow metabolism, and you want to be storing extra fuel, right? And so So to me, all these things are not necessarily, they're not exclusive and they're not opposing. They're all part of. It's just, it's a very complicated process with a lot of different parts, and they're all working together to achieve the outcome, which we call metabolic syndrome, because in our situation, we don't have to store extra fat for winter, and we're not going to actually hibernate. We don't have the option to hibernate. So in our case, it looks like, it looks very bad. But um, just to give one point, so Natambi is one of my favorite researchers, and he's done a lot of the work on SCD1, the desaturase that makes the monounsaturated fat. And what he showed is in mice, if you if you if the mice don't have SCD1, if they can't make monounsaturated fat, all of the inflammatory markers go way, way down. And so NF kappa beta is this very TLR4 is in your is on the gut lining, and TLR4 is the thing that actually recognizes these bacterial sequences in your gut and starts the inflammatory process. If you eliminate SCD1, TLR4 goes down by like 75%. I mean, it goes way, way, way down. If you eliminate SCD1, the NF kappa beta, which is, the oldest, biggest controller of sort of inflammatory pathways, doesn't go into the nucleus and turn on tissue necrosis factor alpha, which is the thing that ultimately causes the release of the CD38, right? And so Natambi actually showed that, so the animals are going into the fall, they're increasing SCD1, that's increasing the production of these monounsaturated fats. That the very increase in monounsaturated fats is leading to increased TLR4 and NF kappa beta, all these immune cells. And if you just eliminate the ability of the delta 9 desaturase, you eliminate that whole inflammatory cascade. And so
is it your speculation that activation of that delta 9 desaturase is one of the primary contributors to inflammation in the body, and what's largely responsible for the elevation in C-reactive protein that most people observe?
Yes, absolutely.
Yeah, and what type of documentations do you have or observations in reductions once that pathway is modulated?
Well, like I say, I mean, I think the strongest evidence that I know of is in mouse models, right, which are imperfect, where you, like I say, if you eliminate the enzymes that can create monounsaturated fats, you almost totally eliminate the inflammatory program, right? So that, I think, is probably the single strongest bit of evidence. But like I said in the last show, all of us, if you look at historical studies of human adipose tissue, 97% of people have more monounsaturated fat and less stearic acid than they did 100 years ago, right? Our whole society has this increased ratio of monounsaturated fat to stearic acid.
And quantitatively, the monounsaturated fat is elevated as opposed to what it was in the past, right?
Right. And
so the oleic acid is elevated.
Right. And so here's the second piece of really good information about this. So I talked about the last time. I sell something called steer oil ethanolamine, right, as a supplement at
otherwise known as SEA.
Also known as SEA on fireinabottle.net/shop. And so what you see in torpor, when you see elevated delta 9 desaturated, elevated SCD1, is that stearic acid, which is the thing that's being converted to oleic acid, monounsaturated fat. So oleic acid goes up and stearic acid goes down because that's what the enzyme does, right? And if your stearic acid levels drop, then your SEA levels drop. And SEA binds directly to NF kappa beta and prevents it from going into the nucleus and starting the nuclear cascade, right? And so as winter approaches, animals upregulate that SCD1, the delta 9 desaturase, and SEA production goes down, and then again that allows NF kappa beta to go into the nucleus to release all these genes. One of those genes is CD38, and CD38 starts breaking down your NAD plus, and you're into reductive stress, right?
So this is SEA. Is it a covalent bond when it's binding to this?
I don't know. It's not a covalent bond.
But is it eliminated, excreted with it intact? Is it excreted through the kidneys or the sweat or?
Yeah, I mean, I think it's broken down and excreted. It's just ethanolamine and stearic acid, which are two very common bodies. And I think there's an enzyme that just dissociates the ethanolamine and stearic acid, and the stearic acid is probably just burned like a normal fat, I imagine.
That sounds intriguing from molecular biological perspective, but why don't you share one of the clinical benefits that most everyone watching this would be intrigued with, because you've had personal experience with it. And I think one of the primary benefits for this specific supplement, I'm a huge fan of supplements, certainly selling enough, but there seems to be a target audience for this specific one because certain magic happens when you're taking it. The magic being your metabolic rate goes through the roof. So why don't you explain that?
Right. And so I noticed, right. And so you can look, we can look at all the studies of mice, right? The SEA reduces inflammation. It reduces production of TNF alpha. It also suppresses SCD1 in the liver of I think it was in rats. It suppresses the delta 9 desaturase in the liver. And so it's doing all of the things that you would predict would help your metabolic rate, right? And so when I started taking it, I was a little unclear at first, right? We were trying a new thing about exactly what dosage, and I started taking 1,200 milligrams a day, and that might have been a little too much because my body temperature, I was taking 600 milligrams for a while, and that was fine. My body temperature was up. I was I was getting into the like 98.4, 98.5, and then I went to 1,200 milligrams a day, and I shot up to like 99 and a half some days in the morning, and that was too much for me because I was like, it was just uncomfortable to be that warm all the time, and I was almost feeling a little flu-like. So I went back down to now I'm doing 300 milligrams twice a day, but that it just, you can see right away with that the increase in body temperature. And I think a lot of people, it's not just me. I mentioned there's the discussion group on Reddit called r/saturated fat. A lot of the people on there have experience with the SEA, and a lot of people have had that same experience with it. And so, and I do believe that, like you say, that experiment in rats where they show that the SEA is blocking that inflammatory pathway, and then when you look at the mice that Natambi worked on and showed that, hey, if you get rid of this desaturase enzyme, stearic acid goes up, oleic acid goes down, and the inflammation goes away, right? And then you look and you say, well, what's happened in humans over the past 100 years? Well, stearic acid has gone down, and oleic acid has somewhat been replaced with linoleic acid, but the ratio of oleic acid to stearic acid in humans has gone from like 8-1, I want to say, in 1940 and 1960, and now it's like 15-1.
Double.
In some populations, I've seen 20-1. Like in clinics where they're doing weight loss surgery, bariatric surgery, the ratio of oleic acid to stearic acid can be 20-1. Whereas if you look at data from 1960, that ratio is typically less than 10. And so that's a huge ratio that has just dramatically changed over the past 100 years. And now you look and everyone has, leaky gut, inflammation, metabolic syndrome. And again, what do the hibernating animals do as winter approaches? Well, they upregulate delta SCD1, delta 9 desaturase. They decrease stearic acid. They increase oleic acid. Inflammation increases. Their gut gets leaky. The CD38 is released. NAD plus levels drop, and they fatten up for winter. Right. And so, And so that's the parallel, and
It's not where we want to go. So
right.
one of the take-home messages for this, I think, is, if you understand molecular biology and nutritional biochemistry, you would appreciate that oleic acid is actually quite common in food supplies, and one that is common, typically viewed. And you've done a video on this, a podcast, actually a video, explaining this. That olive oil is really high in oleic acid, and that's probably why. Oleic and olive, I think there may be some similarity in the etymology of the word, but certainly the other source that I had a lot of, because I don't really particularly care for the taste of olive oil, so I've never really used it that much.
Yeah.
And I thought that it would probably be okay for most people. Just have to be careful, because 80 to 90% of olive oil is adulterated with seed oils. So why would you want to take that? But if you've even found the pure, really expensive one, highest quality, it's still got some linoleic acid in it, but it has a lot, like 2/3 of it is oleic acid. So it's a source of this problematic fatty acid that you're saying has went in the wrong direction. So, and here's where I announce my understanding of this fact and to the world, that I used to embrace macadamia nuts as the only nut that was useful to eat, because it essentially is the only nut that doesn't have much, well, it doesn't have many carbs, which is one of the reasons I initially embraced it. But it also has virtually no linoleic acid. It's almost all oleic acid. And you would think it'd have a little bit more saturated fat because it's a tropical nut, but it's got a mono in there. It's really high. It's almost as high as olive oil, actually.
Yeah.
So, and I like nuts. Who doesn't? Most people like nuts. And, I would buy 10 pounds at a time, and I'd go through it in a month or 6 weeks. And I'm not doing that anymore. I'm off of macadamia nuts, and I'm not recommending them to anyone. I think, yeah, they're neat. They're kind of pricey too. But I think just from a processed food, I mean, have you ever seen, I was in Costa Rica, and there are the scarlet macaws. I think it's scarlet, some type of scarlet bird, beautiful bird, absolutely beautiful. Then they go into the macadamia nut trees, and they get a macadamia nut. It's a big thing. It's like a big, huge apple, and they have to peck away at the thing for like 15 minutes or a half hour just to get one nut.
Yeah.
Just to get one nut.
Well,
It's not easy to get, and yet we eat them by the handfuls. So probably having 1 or 2 nuts a day is not going to hurt you, but having,
yeah.
3 ounces, come on.
I mean, my mother really likes nuts, and she often, when I go home, she'll have a bowl of salted nuts out, and I'll tell you, if I have one handful of those nuts, I can't stop myself. I mean, they're,
Well, there's a number of reasons for that. They salt them and they roast them, and probably in seed oils. But
right. Yeah.
they're not raw nuts. So, but even if you had the raw nuts, it would still be problematic. It's because all of them are high in linoleic acid, except for macadamia, but that's got oleic acid. And I'd like you to elaborate on that, because you're the expert and you study this really well, and you understand. I mean, you've given us the information about the problems of oleic acid, at least in the tissue. But how about dietary oleic acid?
Right. And so this is a little more, right. This is controversial, because everybody says, well, what about the Mediterranean diet, and what about Italy? Yeah. And I still, everybody says the Mediterranean region is so healthy. I did this video called Metabolic Challenge, and it says France, Italy, Spain. And if you look, rural southern Italy is actually one of the most obese places on Earth. The areas where they grow they grow the olives. Greece has a huge wave of childhood obesity. Spain is one of the most diabetic countries on the planet, and Italy is about to overtake the U.S. As having the highest rate of fatty liver. And so, if you look at the countries where olive oil is actually eaten normally, like, they're really not doing well metabolically. Like, if you were going to pick somewhere on the planet to hold up as these guys are doing really well metabolically, right now, given what everything may be wrong with our current place on Earth with the plastics and all the other issues that we have, the Mediterranean region is not doing well. Mm-hmm. I think, I would maybe pick, and so to bring it back to starch, right? Who is doing well? Well, what area of the world has the lowest rate of diabetes is actually West Africa. Mm-hmm. And what they eat in West Africa, the staples in West Africa are starchy tubers. Plantain, which isn't actually a tuber, of course, it's a banana. Um, uh, Manioc, aka, which is also called cassava, and the African yam. These are huge staples. And you know what they also eat, which is very interesting, is one of the main meats consumed in West Africa is beef skin, which, as you know, is all collagen. And so in West Africa, they're eating a lot of starchy tubers, and they're eating a lot of collagenous meats, beef skin, and they do a lot of stews. They have a lot of stews with like cow hooves that are slow simmered to get the collagen out, to get the gelatin out, and that's the basis of the diet. There are starchy tubers and gelatin and collagen, and they have some of those countries have diabetes rates that I think are below than 3%, compared to Spain, which is 11% now, something like that. And so, so I look at that, and that to me seems more like the way to go.
Yeah, I couldn't agree
Rather
more.
starch and gelatin
I have
than
now,
olive oil.
but my idea of the healthiest meal possible would be white rice cooked, and I have about 300 grams twice a day, so it's 600 grams total, with cooked in bone broth, and the bone broth is created from bones, specifically the hip bone of a cow, which is called knuckle bone, and I usually cook 3 of them, and makes enough bone broth for a little bit less than a week. So, but you cook that up in there, and you have the rice, and then you top it off with some egg yolks and maybe a little beef liver, and you got literally probably one of the healthiest meals on the planet.
Yeah, no, I think that's right, and
very high in the collagen proteins, low in the muscle meat. You could have a little muscle meat in there. I have 3 ounces of meat a day, so I could throw that in there, but I'm not gonna have much more than that. But more collagen and some egg yolk. Egg yolks is egg yolk and liver are the 2 most nutritionally dense foods on the planet.
Yeah, agreed.
You're going to get all your vitamins, most all your vitamins from there.
Right, and so then,
I did not say egg whites. I did not say egg whites. I don't eat egg whites. I feed the egg whites to my chickens.
Right. And my only caveat to that is sometimes I prefer the tubers to grains, so I might do something like a cassava or a potato as opposed to the white rice.
They're pretty similar, though, from a starch perspective. I mean, there are different types of starches, there are different structures and stuff.
Yeah, similar. If you're specifically worried about weight loss, I think
the tubers are better.
The tubers, the cassava is very low protein, and so we talked last time about balancing the branched chain amino acids and the glycine, and so rice and corn and wheat are all pretty high in the branched chain amino acids, and the cassava has like almost none, and the plantain has almost none.
Not as high as the other grains. It's relatively low. It's really one of the only grains without linoleic acid. There's virtually none in there.
Right. No, it's very low in linoleic acid. So it's great in all those things, right? It's fine. It's just, for me, I'm trying to go really, really low in BCAAs. And like you say, I eat a little bit of meat just to make sure I'm getting the protein that I need.
Yeah. So
but that's a subtle
one caveat about the cassava, though, and I've got another guest on, Sally Norton, who's done a great job in educating people about the dangers of oxalate, and they should be educated because it's significant. But you know what I've learned? That if you have a healthy microbiome, it's a non-issue. And I'm actually going to have her on to discuss this. You know why it's a non-issue? Because what is oxalate? The molecule. You would know. Do you know what it is?
Not off the top of my head. I should know this.
It's dicarboxylic acid.
Right.
What is that?
Right.
It means it's 2 CO2 molecules bound together. That's all it is. That's right. 2 CO2 molecules. So guess what? If you have a healthy microbiome, that is fuel. They thrive on it. And when they eat it, they turn it into carbon dioxide, which is a nutrient-free gut. It's like perfect. Yeah. If you don't have a healthy microbiome, all bets are off. That thing can destroy your health 100%.
Right. Yeah.
So
100%.
it's
No,
the
I
it's the
agree with you.
microbiome, specifically the oxalobacter bacteria, that digests this, and that's an obligate anaerobe. And if you don't have mitochondrial energy production, you can't create the environment for that bug to thrive in your gut. So the key is the controlling the gut, and then you can eat all the cassava you want, because it's actually fuel for the beneficial bacteria. Not it doesn't feed the pathogenic bacteria, because they're not really growing there, because they competitively inhibited out of existence because of the good guys.
Right. Yeah, no, I think that's 100% right. And just going back to the starch for a second, one of the things about starch is if you look at people when they're eating a starchy meal, and if you can monitor them, and this is without giving too much away, this is kind of the types of things related to this that we sort of want to look at in the lab. But if you look at if you eat a bunch of starch, your NAD plus availability actually increases postprandially. After the meal, you actually have more NAD plus availability if you're burning a lot of starch, right? And so that's where that kind of thinking about talking about more starch based diets, right? It all comes down to reductive stress. And here's a fun one, just thinking about starch and thinking about saturated fat versus unsaturated fats, or specifically monounsaturated fat in this case. So this experiment is done in humans about 4 or 5 times. And what they do is they sit people down and they feed them either, I don't know what the starch source is. It's probably some kind of bread. So it's like bread and butter or bread and olive oil, right? And then they measure how much oxygen they're consuming and how much carbon dioxide they're breathing out, and that's called respiratory exchange ratio. And if you look at those two numbers, you can kind of determine how much carbohydrate people are burning and how much fat people are burning, right? And so if people eat bread with butter, they'll have a relatively high respiratory exchange ratio, and that means that they're burning a lot of the carbohydrate in the meal. So you eat bread and butter, you can burn that carbohydrate cleanly, which is the right way to do it. If you eat bread and butter, you want to burn those carbs first, and then as your kind of blood glucose levels drop, then you'll start to burn more of the fat from the meal, right? That's the correct sequence. If you eat olive oil and bread, you burn less of the glucose right away. That I would argue that the olive oil makes you essentially acutely insulin resistant, because the insulin, you eat that bread, the insulin is signaling, it's trying to clear the other things out to allow that glucose to burn. But if you eat bread and olive oil, your respiratory exchange ratio will be lower, and that means you're burning more fat. And of course, if you read those articles, they say, oh, eureka, if you eat olive oil, you'll burn more fat than if you eat butter. When I look at that, I say, this is bad. This is not what you want. You want
same
to burn
data, different outcome, or different perception of it.
Right. And so I agree 100% with all of those studies. Like, that study's been done 5 different times.
Yeah.
And it's also true, by the way, of the polyunsaturated fats. Either of those fats,
and that's why, because that olive oil, 80 to 90% of the time, is going to be adulterated with polyunsaturated fats, like the PUFAs in the LA. So most of the time, that's what you're going to get. But even if you had the purest one, you still got oleic acid, which is definitely contributing to the problem. You were making the supposition that it was pure, but 80% of the time it won't be, or 90%.
Right, but the oleic acid, when you consume it, it creates OEA, which is sort of the opposite of the SEA that I sell. And that OEA, what it does is it activates another nuclear receptor called PPAR alpha. Mm-hmm. And PPAR alpha is kind of works the opposite of insulin. It actually activates something called pyruvate dehydrogenase kinase, which turns off pyruvate dehydrogenase, and it actually directly turns off glucose metabolism. So every time you eat monounsaturated fat, your body,
olive oil or macadamia nuts,
right? Your body makes a signal that activates PPAR alpha, and the PPAR alpha goes out and it turns off, your ability to break down that glucose. And that might be somewhat just, I mean, if you're eating a meal that's higher in fat, you don't necessarily need to burn as much glucose. But still, if you compare, bread and butter to bread and olive oil, to me, the bread and butter looks like that's what you want to do in the, after a meal, is you want to burn those carbs. Because if you burn those carbs, your NAD plus availability will go up. And I said this in the last show, but there's this old saying called fat burns in the flame of carbohydrate. And you get that NAD plus availability up, and you kind of get that flywheel spinning, and now you can efficiently burn, the fat in the meal and also your stored body fat, because the carbs are really driving that process.
Yeah. So one of the ways you can do that we talked about earlier is this SEA supplement that will increase your metabolic rate, which is exactly what you want to do. The whole key focus, and I've recently come to appreciate this, is you want to increase your body's ability to create cellular energy, which means increasing your metabolic rate, which means you have improved mitochondrial function, and you go from mitochondrial dysfunction to exemplary mitochondrial function. And that's the whole purpose. If you can do that, it doesn't matter what disease you have. It doesn't matter what label you've been given. It doesn't matter what drugs you're on. If you can get your system to make the energy it was designed to make, most every disease is going to start to reverse and get better. You can get off your medications. So this is why it's so critical to have this as focus. Now, you can inexpensively measure your temperature. That's a very powerful way. We have very accurate, inexpensive digital thermometers that you can measure within a tenth of a degree what your temperature is. As long as you weren't sucking an ice cube, you're going to get a pretty accurate measurement, right? So
yeah, and if people want to go out and get this is actually important. It's sort of silly, but if you want to get a good digital thermometer at your local drugstore, so they have these instant read thermometers, and those are not nearly as accurate. They give you they give you an answer within like 10 seconds, but that answer is usually not very accurate. You want the old-fashioned slow read thermometer that takes like a full minute to give you a temperature. Those are the most accurate. They're also the cheapest. Yeah. So you don't want to buy the $20 thermometer. You want to buy the $10 thermometer. Yeah. The old-fashioned one. Those are the best.
Yeah. So that's key. And if you want to take it to the next level, and one of the reasons I'm so passionate about connecting with Brad is that we are developing a lab test. And I'm going to let you take it from here, but essentially it's 3 redox pairs. Actually, it's 4 now. No, it's 3. It's 3. Lactate and pyruvate. It's acetoacetate and beta hydroxybutyrate and oxidized glutathione and reduced glutathione. Those are 3 redox pairs. And by evaluating them, you can figure out what your redox status is, which is probably likely, and I'll let you discuss this, even more accurate than measuring NAD plus directly. And I speculate that we're going to innovate this, and there's a lot of companies that are going to start offering this. And we're going to offer this test because this is a test that you can get in the not too distant future. We're going to make it available because we're having a whole system where I'm hiring health coaches, training them, and the health coaches are going to facilitate your ability to enter into a clinical setting inexpensively. It's not going to cost an arm and a leg. It'll be relatively inexpensive, more affordable than maybe your copay to see your doctor. And you can do these tests. That doesn't include the test, but these tests are like, it'd be well over $1,000, maybe even $2,000 if in a lab like LabCorp or Quest, right? What we're doing, but we're going to probably do it for like a lot less than that, like maybe 1/6 or 1/10 of the price. So because it has to be affordable or we can't do it. And the information you're going to get is going to be literally some of the most precise measurements of mitochondrial function that you could possibly be out there. And this way you can monitor yourself and along with your symptoms and your temperature and other things and let you know that you're on the right track. So why don't you take it from there? Because I think we're excited about this because we're going to do a lot of clinical research. We're going to put people on innovative protocols and actually measure what happens to their biology very accurately and precisely and reproducible.
Right. And so this is kind of an old idea that we've known since the 1960s that if you look at your lactate versus pyruvate, there's an enzyme called lactate dehydrogenase which is in your cell, and is basically equilibrating the lactate and pyruvate based on how much NADH and how much NAD plus it has. And so that lactate pyruvate ratio is an indirect reading of your cytoplasmic redox balance, but it's a very accurate one. And NAD plus and NADH themselves are very hard to measure directly, and they're if you try to collect serum, they're probably going to they could probably change back and forth very rapidly. So like you say, it's unclear how, they're kind of a moving target, but the lactate and the pyruvate are a little more stable. That gives us a reading. And then the other one is your ketones, your acetoacetate and your beta hydroxybutyrate. The same thing happens in your mitochondria. There's a mitochondrial enzyme that is swapping the two back and forth based on the amount of NADH and NAD plus that it has. And so those two readings give you kind of an idea of your whole body redox balance. And you can see separately in the cytoplasm and in the mitochondria. And both are important because in the mitochondria, of course, you need NAD plus just to drive your metabolic rate forward. Every time you're burning food, you're burning acetyl-CoA, you need NAD plus, and you can't have too much NADH or the whole system just kind of bogs down. In the cytoplasm, I've talked a lot about these desaturases and how they lead to all these feedback loops with the activating the oxidized PUFA, activating the nuclear receptors, etc. Well, those desaturase reactions happen in the cytoplasm. So if you have high NADH to NAD plus in the cytoplasm, you're going to push forward the activity levels of those desaturase enzymes. Also, the cytoplasm is where you do de novo lipogenesis, and so that's where you make fat, and that process is also driven by reductive stress. And so the two compartments are both important and interesting in their own way, and we can tell them separately. And then the third one is glutathione. Now that one.
Well, before we go to glutathione,
yeah,
the question on the two compartments, intracellular versus mitochondrial. I would intuitively think that if there's reductive stress, it's going to be in both compartments, and they both should be abnormal. But are there clinical conditions where it's only abnormal in one compartment?
Yes, there are, and so what it looks to me like what can happen is you can start with reductive stress in the mitochondria, and if it gets too backed up, then you can have this huge release of superoxide and reactive oxygen species, and these oxidative things can be released from, like, once you're into like complete frank metabolic dysfunction, the mitochondria starts releasing all these reactive oxygen species, which in a controlled situation, these reactive oxygen species are very beneficial. Mm-hmm. But if the mitochondria gets too reduced, you start releasing all these reactive oxygen species, and that can actually wind up oxidizing the cytoplasm to some extent. And so you can see paradoxical situations where one is reduced and the other is oxidized, or the other way around. I think, generally speaking, I would expect to see them both be.
And if they weren't both, most likely the mitochondrial would be the one that's in the reduced state, and the cytoplasm hasn't been impacted yet. Is that most likely?
Yes, I think that's right. So
it's an earlier warning signal of mitochondrial dysfunction. If you've got both compartments trashed, you're in deep weeds.
Yeah, I think that's right.
Okay. So I interrupt you.
And it's also, and what we should also say is it's possible. So glutathione, but then to bring glutathione in it. So glutathione is the best measurement of actually. So the NADH is taking electrons from acetyl-CoA. It's taking electrons from your fuel. Mm-hmm. And if your metabolic rate slows down, that NADH kind of piles up. And so the NADH shows you specifically how fuel is moving through the mitochondria, and in the cytoplasm, the NADH kind of tells you how well glycolysis is working, or if you're bringing in glucose and you're converting into pyruvate, that affects NADH and NAD plus. That's interesting. But then in the mitochondria, or sorry, in the cytoplasm, the thing that deals with mopping up these reactive oxygen species is glutathione. And so the glutathione almost tells you more how much how much hydrogen peroxide is being released from the mitochondria, and the glutathione is being used to kind of mop that up. So if your glutathione pool is very oxidized, that suggests that you're actually creating a lot of reactive oxygen species. And so all of these different redox pairs tell a little bit different story.
So help me understand this, because it's counterintuitive to think that the reduced version is beneficial, is not beneficial, because normally an antioxidant, so a reduced molecule is not an antioxidant, right?
Right.
Antioxidants are oxidizing agents. So, which is what glutathione is. It's an oxidizing agent. So, but the knowledge that you're bringing forward, and many others like you, is that the reduced form is not what you want. You want the oxidized version, especially NAD and pyruvate and acetoacetate. You want those oxidized. But it seems like it's not the case for glutathione, because it's sort of mopping up the damage. Is that correct?
Right. And so this is the kind of confusing thing when we start talking about antioxidants.
Yeah.
You have this initial, I don't want to call it damage. You have this initial reaction. Mm-hmm. What happens is you have too many electrons. One of them jumps onto oxygen. And we talked about this in the last episode. We talked about too much NADH at complex 1. And what happens is the electrons kind of bounce out and they jump onto oxygen and it forms superoxide.
And they jump because there's a block in the electron transport chain. They cannot seamlessly go down that chain. There's a block somewhere.
There's a block, or there's just simply too much NADH. Too much fuel is pouring in, and you don't have the kind of energy demands.
Couldn't you make an argument that if you had really exemplary mitochondrial function, you almost can't get enough fuel? It'll just go all the way through.
I think there are situations where you can just literally overwhelm. The last time we talked about the carburetor example, where you need to balance oxygen and fuel. Right. I think you can overwhelm the mitochondria. If it all depends on how fast you're bringing in all the different fuels, right? And obviously mitochondrial complex, it's very complicated, right? Because that has to do with how fast you're bringing in fats. It has to do with the Randall cycle, and it has to do with branch chain amino acids.
In most normal digestive processes, if you're eating rationally, you're not doing some superhuman feat like eat 100 hot dogs in 10 minutes or something. You're likely not going to exceed that capacity.
Well, I think you can look at traditional. I mean, you can look at America in 1940, right? 1960, which was, we were a very well-fed country. People ate tons of calories back then. I posted the 1939. It said, according to the USDA, in wealthy households, an adult man was eating 4,500 calories a day in 1939, according to the USDA.
A lot of calories.
And so people were eating a lot of calories back then, and yet we saw very little of these problems, right? Mm-hmm. And so I think that the increase in unsaturated fats, like I say, what it's doing is it's causing this. The fat is jumping the line in front of carbohydrates in our mitochondria. And so that's suppressing carbohydrate metabolism. And when you suppress that carbohydrate metabolism, you lower the NAD plus availability. And then instead of burning the glucose oxidatively, you shift over to doing de novo lipogenesis. You shift over to.
But in that case that you're describing, that's not a surplus fuel. That's the wrong fuel.
Right, it's the wrong fuel. Yes.
Yeah. So
yes.
What I was thinking is surplus. If you had the right fuel and you put it in there, it's hard to eat too many carbohydrates, like starch, and back 100%. It up. Right. So
yes,
you could double that. So it's not the quantity of fuel; it's the type of fuel that.
It's the quality of fuel.
Metabolically can derange your system.
I agree with that. Yeah. I agree with that. Yeah. Yes. And just to go back to the point about the glutathione, so in the antioxidants, right? So the antioxidants are paradoxical because the antioxidants are electron donors. They have this electron to give, right? Mm-hmm. And so if they give that if they give their electron to NAD plus, like NAD plus is an electron acceptor. So if the antioxidants give electrons to something like NAD plus, they can actually cause reductive stress,
Yeah,
right?
absolutely. That's the
Because
last
they're
thing you want to
donating
do.
electrons. But on the other
Counterproductive.
hand,
Counterproductive.
once you have something like superoxide, which has an unpaired electron, or once you have um, a lipid peroxide, which is a free radical version of a fat, which are typically the first things that are polyunsaturated fats will get damaged, and they pick up an extra electron. Then an antioxidant like vitamin C can come along and hand it an extra electron, and now it has a pair of electrons, and it's no longer a free radical. It no longer has that unpaired electron, right? And so the irony of that is that it's the donation of the extra electron from the antioxidant that caused the free radical in the first place. And then it's a second donation from the antioxidant that diffused the situation by giving it the pair of electrons, which is what it really wants, right? And so antioxidants are on both sides of this equation. And you look at glutathione, we want reduced glutathione because reduced glutathione is used by our enzymes to get rid of things like lipid peroxides and to get rid of things like damaged proteins, etc. But on the other hand, cancer cells are loaded with reduced glutathione because cancer cells are in severe reductive stress. And one of the ways you can actually store extra electrons and store reductive stress is by building a huge pool of reduced glutathione. And so glutathione is acting as an antioxidant, and to prevent lipid peroxides, you want to have a mostly reduced glutathione pool, but at the same time, it's a slippery slope, and you don't want to have a buildup of glutathione. You want to have kind of the right amount of glutathione.
Yeah, the sweet spot, the Goldilocks dose. You know,
right.
You had mentioned lipid peroxides, and I wonder if you could. It confused me a little bit because I was thinking of lipid aldehydes, reactive aldehydes, which like malondialdehyde or 4-hydroxynonenal, both reactive aldehydes. So what's the difference between an aldehyde, a lipid aldehyde, and a lipid peroxide?
Yeah, so it's I mean, it's essentially they're very equatable. I mean, it's
they're both are they both oxalams?
They're both right. They're both oxidized linoleic acid. Oxalam just means oxidized linoleic acid metabolite, and so you can get aldehydes from linoleic acid. You can also get them from other polyunsaturated fats, right? And aldehydes often happen in cooking oil. So as fryer oil breaks down, it creates a lot of aldehydes, and that sort of is when the one of those double bonds actually breaks. And so MDA are these linoleic acid is like 18 carbons long, and it's got the 2 double bonds. And what will happen is one of those double bonds, or maybe it's right below the double bond. I'd have to look at the biochemistry. I don't have it all memorized in my head, but that bond actually breaks. And then you get this short little fat with this reactive group on one end, and that's an aldehyde. And so you have this like 18 carbon chain, and it breaks into like a 14 carbon chain and a 4 carbon chain, and they're both kind of reactive.
That's the difference. Okay.
Right, as opposed to a lipid peroxide, which is the full-on lipid, so it's still linoleic
so the
acid.
aldehyde is broken down. That's the difference. It's metabolized.
Right, it hasn't broken all the way down into an aldehyde yet. Is probably the easiest way to say that.
Okay. Yeah, thanks for helping that. I did, because I really never studied the metabolism of the oxalams. I just know they exist, and there are many of them. There's dozens, if not hundreds, I believe.
Right, and I think it's very important to think about them. We sort of have 2 different pools of them. We have things like aldehydes. The aldehydes happen because of spontaneous damage to the fats, right? You've generated too many reactive oxygen species or whatever has happened. The fats have oxidized and they've broken down in natural ways that don't require enzymes.
Mhm.
And then you have these enzyme systems, and I keep coming back to 12 heat and 5 heat because uh, 12 heat and 5 heat are specifically made from cytochrome P450 1B1, whose expression is controlled by the aryl hydrocarbon receptor, right? And so it's like, uh, if you see a lot of 5 heat and 12 heat, like they're not there by accident. They're there because we made an enzyme that produces them. Like you don't, if you just have spontaneous oxidation, you don't wind up with a lot of 5 heat and 12 heat and 15 heat. Like those are there on purpose because usually because of upregulated AHR activity is why we have those, right? Those don't just spontaneously happen. And there's a lot of other I bring those up because they're the most tightly associated with heart disease and they're the most tightly associated with obesity and diabetes. Um, and they're, but it's not to say that the aldehydes are good. Obviously, you don't want MDA buildup. That causes all kinds of other problems, and they can cause, mitochondrial damage as well. Um, but there are sort of two separate programs happening, right? One is just kind of what we think of as oxidative stress, where it starts with reductive stress. It starts with this buildup of electrons. These electrons get released in the electron transport chain. This winds up damaging these fats. The fats go out and they can do damage to the enzymes that are involved in the mitochondria, and you just have this kind of like gradual decline in mitochondrial function. And the other pathway is more of a targeted pathway that is driven by these, nuclear receptors, and it's driven by cytochrome P450 enzymes. And that's all very regulated by our enzyme systems, and that's regulated by and that's involves the desaturase enzymes and PPAR alpha and the aryl hydrocarbon receptor, and that is sort of like its whole own separate loop, if that makes sense.
Yeah, that's important stuff. So I'm so excited that you transitioned to research. That's your true passion. It's one of the ways that you excel in life is to identify what gives you joy and be able to facilitate your choices to allow that to happen. So congratulations on that. And
Thank you.
so we're really excited to collaborate together and, bring more joy to the world by some of the findings that you help elucidate and some of the tools you'll create to allow people to utilize to help guide them to make better choices.
Yes, it's very exciting, and thank you for giving me the opportunity. You know, it was. I'll say that I've really enjoyed having the opportunity the last few years to really just dig in the literature and kind of catch up. And you mentioned the possibility of doing some research, and then I realized how much I actually really did want to get back in the lab do
Yeah,
that research. So
yeah,
thank you for sort of reigniting that
you're
flame in
welcome.
me, because
Yeah.
I'm really excited now that I've caught up. I've caught up to now what's happened in the literature, and now I'm ready to push forward. Let's go. Let's go get them.
That is not an easy thing to do, folks. It requires a special type of brain, one that is actually producing significant energy. And it's important to understand this because the brain only consists of what by weight is 2% of your body weight, yet it consumes 20% of the energy of your body. 20%.
Yeah.
The heart isn't far behind, actually. It's pretty close. And I think collectively it actually consumes more energy denser wise because it doesn't weigh as much, but it consumes proportionally more. Which is your heart and your brain are really metabolically active. So that's one of the reasons why we have such an epidemic of heart failure, because there's not enough cellular energy. And we have brain failure like Alzheimer's. Yes, there's amyloid beta plaque, but why is that plaque there to begin with? Well, maybe because there's not enough energy to fold your proteins the right way, right?
Yeah, and one thing I know about the heart. So a lot of the interesting things that we've learned about this reductive stress causing oxidative stress has happened in the heart in these they call them ischemia perfusion experiments, because they're trying to simulate a heart attack.
Yeah.
And they
Or stroke would be the same thing too.
or a stroke, right? Same thing,
stroke.
right?
Yeah.
And so what they can do in a mouse or a rat is they can shut off the flow of blood to the heart for half an hour or something, and that's the ischemia, and then they open it up and they let the blood flow back in, and that's the reperfusion. And what you see is that when the blood flow is cut off to the heart, and of course the oxygen is cut off, and now the electrons in the mitochondria are ultimately flowing back to oxygen, right? And that gets released as CO2. And so when you cut off the blood flow, there's no oxygen, the electrons can't flow back. Well, what happens? NADH levels build up and build up and build up and build up because there's nowhere for the electrons to go. So all of the electrons are kept in this pool of NADH, right? So cutting off oxygen puts the heart into heavy reductive stress. Then when they release the blood and the blood hits the heart and the oxygen hits the heart, it lets off this huge wave of reactive oxygen species, because now you have all of these electrons and they're built up in that NADH pool, and the oxygen comes in and those electrons just jump onto that oxygen and it makes superoxide, and you have this huge oxidative wave. And so that's one of the main ways that we know that the root cause of oxidative stress is actually reductive stress and the buildup of electrons in this NADH form.
Yeah, and it's interesting the situation you described, because there are two nutraceuticals that I don't recommend using routinely, but in this scenario, they are life-saving. And one is melatonin, and the other is methylene blue, both of which are antioxidants. Methylene blue specifically is a very potent oxidizer. So if you put them at significant levels and you've gotten the exposure, you've got that ischemia reperfusion potential. In other words, you have an ischemic event and there's not you're not getting perfusion of the blood, and you know you're going to get it soon for whatever reason. That's when you load those things on board, and you can reduce like 90% of that injury with those oxidants, right? Because you're sucking up those electrons with the methylene blue and the melatonin, and you're not allowing them to go to oxygen. Those electrons are only a problem when they hit oxygen. They're not a problem when they hit an antioxidant like methylene blue. It's fine. It's no problem at all. But
right, except like you say, methylene blue is really an oxidant. And so it is. One of my favorite supplements that I do take is alpha lipoic acid. And if you read any of the literature, it will say alpha lipoic acid is a powerful antioxidant. And then if you look at how it works, well, it actually is, you take it in the oxidized state, and it is an oxidant, and NADH will give its electrons to lipoic acid. So lipoic acid is absolutely functioning as an oxidant. It's oxidizing the NADH pool. And the downstream effect of that is the elimination of the oxidative stress, right? But the reason that the oxidative stress is gone is that you've oxidized the NADH pool. And that seems counterintuitive to people. They think, well, you need antioxidants. But it's actually the opposite. You actually need oxidants to get rid of the oxidative stress.
Well, the reductive stress, which causes the oxidative stress.
Right, exactly.
Yeah, but it's definitely there. It's somewhat confusing, but it really doesn't have to be that way. And Brad did a really great job last podcast to interview to help understand it, because it's really simple concepts, but it's confusing if you let your head spin for a bit.
Yeah, you have to actually.
But when understanding it is really important to really implementing right choices to make sure things are not in this reduced state, because that's what causes the problem. That's what literally is the cause of almost all this disease. It's this overabundance of electrons that are unable to find their way to rest comfortably with oxygen and form water. But if they get sabotaged and form singlet oxygen as superoxide and it builds up, you've got massive problems. And it's just to the races. It's just not bad, not good, not a good outcome. And it leaks into the cytoplasm, and you have oxidative stress there. So if people want to find more about you, you've got a podcast on YouTube, or a channel, more specifically, Fire in a Bottle, which is really great. I listen to, I walk on the beach pretty much every day. Most of the time these days, I'm talking to people because I have limited time to talk. So that's when I do my talking. But if I don't have a phone schedule, I'm listening to your podcast because it's really good. I really enjoy it. It's just, it stimulates, it reminds you of different things, because frequently it's difficult to keep all the molecular biological pathways, and it's so good to be reminded. You do such a good job of it. So great podcast. And you also have a site where you sell the SEA, which is, something to consider, if you want to increase your metabolic rate, and maybe you're not getting good results with the thyroid. Not that you shouldn't take thyroid. I think everyone benefits from taking thyroid, but this is another way to hit it at a more fundamental molecular biological level that could tend to dissipate one of the drains on your metabolic rate, which is this overproduction of the monounsaturated fats, oleic acid specifically.
Yeah, right. It helps you. It sort of is the like I say. I think the oleic acid and the stearic acid are kind of the yin and yang of your metabolism, and that SCD1, that delta 9 desaturase, is the thing that kind of decides the winners and losers. And so the SEA is sort of giving you your body the impression that it's that you're in the tropics. Winter's not coming. There's no need to fatten up, and it will like I say, it binds directly to NF kappa beta, and it prevents that whole inflammatory process. It suppresses the delta 9 desaturase in the liver, and it actually helps you break down the OEA and those other endocannabinoids, which are triggering your cannabinoid receptors and giving you the munchies and all of these other problems. And so
that can be side effects from linoleic acid too. It's clearly a mechanism of how it works.
Yeah, absolutely. And so, and it's just, there's just a whole, obviously this whole positive feedback loop is very, it's complicated, but I do think that the SEA is one of the major kind of high level controllers of how.
Where can they get that at your site? What's your site name?
It is fireinabottle.net. Go to fireinabottle.net/shop, and you can get the SEA there. I also have another interesting one there that I think people is worth trying, is something called theobromine, which comes from pu'er tea extract. And this is a highly oxidized tea. They make it in China. It's a very old product, and they ferment it, and it causes this large pigment called theobromine. And theobromine, when in all kinds of things,
it's not theobromine, which is in tea. This is different, or chocolate.
No. It's not theobromine. It's called brownin, and it's just a tea. It's just the brown part of pu'er tea. But we could see that pu'er tea has all these really good outcomes. There's a really good study about how it increases bile acid excretion. It increases metabolic. So we talk about, here's a good example, is this pu'er tea experiment in mice. So they put mice on a high fat diet, and one of them got the pu'er tea extract and one of them did not. And even though the mice ate, I think they ate more calories, the ones that had the pu'er tea, and they remained very lean. And the ones that did not get the pu'er tea became obese. And that study is one of the most distinct studies in mice that shows you that, calories in, right? Calories in don't necessarily make you fat, because those mice given the pu'er tea extract were eating more calories, and they were the ones who stayed lean. And so it really is about metabolic rate, and it's about throughput. And it was very unclear sort of what the pu'er tea extract did. And I think there was a paper that came out in 2023, and what it showed was, remember I said the receptor on the inside of your intestine that recognizes those bacterial signals and starts that inflammatory process of the NF kappa beta. The pu'er tea, it seems like it binds to that TL4, but it doesn't activate it. So it essentially blocks that interaction between the bacteria and the TL4, which begins that inflammatory process. And so, I've been offering a gut health bundle, which is the SEA and the theobromine tea extract together. That is
fireinabottle.net.
Yep. So and the idea of that is just block that whole inflammatory pathway, and like we talked about, the signaling out of your gut microbiome, etc.
All right. Well, after you read the next few thousand articles, we'll have you back on and give us the new insights that you get. I don't know anyone who's read more articles than you, so that's a good thing.
Yeah, well, I hope the information we present isn't too overwhelming. I know it's a lot, but I try to explain it in ways I think people can understand. Yeah,
you've always got your podcast too, so that's another resource.
Yeah, check out the YouTube channel. There's articles on fireinaBottle.net, and everybody should go over to Twitter if you're interested in these new dietary ideas, and a great resource. There's tons of people over there doing experiments. They're talking about it all the time is the Reddit group r/saturated fat. There's some really, really smart people over there. It's a great community, and it's getting better all the time. And I'm constantly amazed when I go over there and see the level of intelligence in the responses.
That's good.
It's really a dynamite resource.
All right. Well, you keep up the good work, Brad.
Well, thank you.
Thanks so much for watching. Remember, hit the like and subscribe button so you can get more videos that can help you and your family take control of your health.